Okay, so we're going to be looking at structure of biopolymers, carbohydrates. Basically, we know that within the cell, small organic molecules join together to form larger molecules. And these macromolecules are those macromolecules of life confound the cell most of its functions. Macromolecules are known to be large molecules composed of thousands of covalently connected atoms. Most macromolecules are polymers. They're built from monomers. Example of such are macromolecules that we know that we find in cells are carbohydrates, the proteins, and nucleic acids. Carbohydrates from small monomers of monosaccharides. Proteins from small monomers of amino acids, and nucleic acids from monomers of nucleotides. The monomers form larger molecules by their condensation reactions accompanied by a dehydration. So to form the larger molecules, while to hydrolyzing the polymers can produce monomers of monosaccharides. This is a diagram depicting how small monomers come together accompanied by a dehydration reaction with the loss of one water molecule to form covalent linkages, while the larger polymers can be broken down with the aid of water in a reaction, in a hydrolytic reaction, which is degradative in nature. Macromolecules can be found in different kinds of species, and the variety of polymers can be built from small set of monomers. They vary from species to species. Even within species too, there's so much a variety. The roles of carbohydrates. Carbohydrates are known to be major energy source for living organisms. They are also means of transporting energy. They are equally structural materials too. They are found in the cell wall of plants. They are found in the exoskeleton of arthropods, insects, crabs, and the likes. They are also found as building blocks of nucleotides. So they are precursors of for other biomolecules, as we already know. Ribose sugar, for instance, is a carbohydrate, is a monosaccharide that can be used in the synthesis of purine and pyrimidine nucleotides. Now, carbohydrates are the most abundant biomolecules in nature. They have a direct link between trapping the energy from solar energy into chemical bonds via which living organisms would produce energy. Now, they are also source of rapid energy production. The stored energy inherent in them can be used as a quick source of rapid energy via their breakdown through several degradative pathways, such as the glycolytic pathway that would yield ATP, as well as some other amphibolic intermediates. They are also structural building blocks of cells. We know very well that some of these carbohydrates, such as the cellulose, are building blocks of cells. Now, the components of several metabolic pathways as well. We find them in pathways for synthesis of other biomolecules, such as glycerol. You find it in lipid biosynthesis. You find ribose in synthesis of purine, pyrimidine synthesis reaction. Some of these carbohydrates are also recognition molecules. They are there for cell-to-cell binding. They form part of the signal molecules that would be recognized on the surface of different cells. Carbohydrates are polyhydroxyaldehyde or polyhydroxy ketones. They are compounds that contain carbon, hydrogen, and oxygen. They are referred to as the hydrates of carbon. They consist of sugars, and these sugars are compounds that contain alcohol as well as carbonyl functional group. Some sugars have the aldehyde group in them. Those such sugars are referred to as aldose, while some have the ketone group, and those sort of sugars are referred to as the ketose. The aldehyde, the ones having the aldehyde group are those with the terminal carbonyl group, while where the carbonyl is found on another carbon chain other than the terminal carbonyl group, it is referred to as what? As a keto sugar. The So basically, the simplest sugar molecules are glucose, while the smallest is the glyceraldehyde. The smallest in terms of size is the triose, is a sugar that can still exhibit all the characteristics of carbohydrates. Glyceraldehyde is an aldotriose. It has a functional isomer, which is the dihydroxyacetone. That is a ketotriose. So a triose in the sense that it has just 3 carbon atoms, while the glucose molecule, which is the simplest, and we call it the simplest because it is that simple form that is found in most carbohydrates. It is also the form in which sugar molecules are absorbed finally into the tissues. Now, the glucose is an aldohexose. That's 6 carbon, and mostly exists in the aldehyde form. The ribose sugars, as well as the 2-deoxyribose that we find in ribonucleic acid RNA and deoxyribonucleic acid DNA, they are 5 carbon sugars, and they differ in the absence of an oxygen on carbon 2 of the ribose to give the 2-deoxy-D-ribose. There are aldopentoses in the sense that they are aldehyde groups with 5 carbon sugars. Classification of carbohydrates. Carbohydrates can be classified into three major classes. One is the monosaccharide, the simpler form that makes up the other forms. Oligosaccharides are short chain carbohydrates, while polysaccharides are those long chain carbohydrates made up of several thousands of monomers of smaller monosaccharides. Now, there are also another group which are referred to as glycoconjugates, with having, apart from carbohydrate moiety, other groups such as lipids and proteins. The monosaccharides, they have some properties. Some of their properties include their being colorless and their being crystalline solids. They are soluble in water but very insoluble in nonpolar solvents. One of the carbon atoms in double bond is double bonded to an oxygen atom to form a carbonyl. The carbonyl group, when it is found at the terminal carbon or the first carbon, is referred to as, the carbohydrate is referred to as an aldehyde or aldose, while where it is found on other carbon atoms other than the carbon one, it is regarded to as a ketose with a keto group. So they are classified also according to the number of carbon atoms they contain. The aldehyde with 4 carbon groups are referred to as tetrosis, while the ketosis with 4 carbon groups are referred to as tetrulose. 5 carbon pentose with a keto group, it is referred to as pentulose, and on. So the monosaccharides, okay, so the monosaccharides, you have the 3-carbon glyceraldehyde, you have the 4-carbon erythrols and trios, you have the 5-carbon ribose and arabinose, 6-carbon mannose, glucose, glucose, and the rest of them. A ketotriose, the glyceraldehyde is an aldotriose, while um, its keto form is the dihydroxyacetone, which is a ketotriose. So the erythrose is an aldotetrose. Its keto form is the erythrulose. And now the monosaccharides, have have the ability to exist as isomers, whereby they have the same chemical formulas but different structures. Their conformation, their spatial arrangement, the spatial arrangement of substituent groups confers on it what you refer to as the stereoisomeric forms. Now, chiral centers are found to be asymmetric carbons that are found within sugars. The chiral centers, the amount of chiral centers is related to the number of possible stereoisomeric forms that could be obtained. Enantiomers are mirror images of stereoisomers. Mirror images in the sense that they're non-superimposable mirror images. The simplest aldose, glyceraldehyde, for instance, has 3 carbon atoms, and the 3 carbon atoms, as shown here, the 3 carbon atoms it's an aldehyde with a terminal carbonyl C double bond O and an hydrogen group attached. It is not an asymmetric um, carbon because it is not bonded to 4 different groups. The 3rd carbon too is also not an asymmetric carbon because it is not bonded to 4 different groups. You can see here it is bonded to 2 hydrogen groups, while the 2nd carbon is the only asymmetric carbon. So this is the chiral carbon in the glyceraldehyde. Orientation, because of its asymmetric nature, it is able to rotate the plane of polarized light either to the right or to the left. When it rotates it to the left, it is shown here, it is the L-glyceraldehyde form, and when it rotates the plane of polarized light to the right, it is called the D-glyceraldehyde. Now, the projection shown here is just the Fisher projection, where the bonds are shown as simple bonds. Now, the simplest aldose, glyceraldehyde, contains 1 chiral center and it has 2 different optical isomers. The amount of the number of chiral centers, like I said earlier, is proportional to the number of optical or stereo isomers that will be found in a sugar molecule. So a simple calculation of 2 raised to power n, where n is the number of chiral centers, is used to calculate for the maximum number of stereoisomers that can be obtained from a sugar molecule. So if a sugar molecule has just 1 chiral center, like we have in the glyceraldehyde, it has 2 raised to power 1 possible stereoisomeric forms, which is 2. So the 2 stereoisomeric forms are shown, the D-glyceraldehyde and L-glyceraldehyde. Now, these 2 are non-superimposable. As you can see, they are mirror images of each other. One has the hydroxyl group to its right, while the other one has the hydroxyl group to its left. So they are non-superimposable, and they are also referred to as enantiomers. They are stereoisomers because of the difference in the spatial orientation of the hydroxyl group around carbon 2. Now, the D and L enantiomers, if you have an equal mixture of these enantiomers, an equal mixture of them, because they rotate the plane of polarized light either to the right or to the left to equal degrees. So if you place them in a container, if you place them in a solution where the two are of equal concentration, they will not be able to rotate the plane of polarized light either to the right or to the left. You just have a 0 degree rotation, hence no rotation. So the cyclic structure of monosaccharides, most of these monosaccharides shown as straight chain oftentimes exist in aqueous solution as in the cyclic form. They predominantly occur in the cyclic ring structures, either as the pyran or the furan ring. The pyran ring is a six-membered ring, as would be shown here, the glucopyranose. Now the furan ring is not shown here, but it is the five-membered ring. This is the furan ring here. So this is also another basis for isomerism, where 5 to 6, 4, 5, 6 carbon sugars and above are able to form ringed structures in aqueous solution. So the furan ring here, and then the pyran ring is 6-membered, while the furan is 5-membered. Now the glucose majorly exists in the pyranose form, while fructose here exists mostly in the furanose form. The ability of a sugar to form the cyclic ringed shape is due to the reaction between the carbonyl group here and the hydroxyl group. The carbonyl carbon, that's the first carbon atom in the aldehyde, and an hydroxyl group in any of these other carbon atoms along the chain results in two different forms, two possible different forms, which are referred to as anomers: the alpha glucopyranose form or the beta glucopyranose form. In the alpha anomer, the hydroxyl group around carbon 1 is oriented downwards, or its position downward, while in the beta pyranose form, the hydroxyl group is oriented upwards. These are two anomers that are a direct result of the rotation that occurs when the cyclic forms are being obtained in aqueous solution. So these anomers are sugar molecules that are different in the orientation of hydroxyl group around carbon 1. Carbon 1 here is referred to as the anomeric carbon atom, and the phenomenon itself is anomerism. It's a type of isomerism. Anomerism whereby mutarotation, the cyclization around between the carbonyl in carbon 1 and the hydroxyl group representing an alcohol on carbon 5 gives the orientation, the two different possible orientation, the alpha orientation or the beta orientation. So these are the alpha and beta anomers, and the isomers that have been shown are the pyran-furan isomer, the functional group isomers of aldose, ketose. Now there's also another isomerism referred to as the epimerism. This refers to sugar whose configuration differ around just one specific carbon atom. Glucose, for instance, is an epimer of galactose in due to the change in orientation around carbon 4 of glucose. Um, if the hydroxyl around carbon 4 of glucose is oriented upward and not downward, it becomes galactose. And these are two different monosaccharides. Now, around carbon 2, if there is an epimerism around carbon 2 of glucose, what is obtained is mannose. So the epimer, so the two groups are referred to as epimers. Enantiomers, on the other hand, are mirror images of each other, as you have in the D and L glyceraldehyde. Now, cyclization of sugars into the pyranose ring or the furanose ring results in the formation of the pyran-furan isomerism. The important reactions in monosaccharides are as follows. The multi-rotation we just observed, where alpha and beta anomers of sugars are formed due to the interconversion when the sugar molecules are dissolved in water. Now, most of the monosaccharides, most all monosaccharides are able to undergo oxidation reactions. They are able to oxidize. They're able to reduce cupric ions and ferric ions into the cuprous or ferrous forms. So in the presence of oxidizing agents, the ions undergo several oxidation reactions. The monosaccharides undergo several oxidation reactions. So oxidation of the aldehyde group in a sugar yields aldonic acid. However, oxidation of the terminal CH2OH alcohol group yields a uronic acid. So for glucose, if the aldehyde group is oxidized, you get a gluconic acid, that is an aldonic acid, while if the alcohol group at carbon 6 is oxidized, it yields a uronic acid such as glucuronic acid. And so if both the aldehyde and the alcohol group are oxidized, it yields an aldaric acid. The carbonyl groups in both aldonic and uronic acid can react with hydroxyl groups in the same molecule to form a cyclic ester lactone. Now, reduction of the aldehydes and keto groups of monosaccharides can also yield sugar alcohol such as alditors. A very common example is the sorbitol, which is used commercially in processing food and chemicals. Now, isomerization reactions such as the epimerization we observed on carbon 2 and carbon 4 that yields mannose and galactose respectively from glucose are also one of such important reactions that exist in sugars. Esterification, the free hydroxyl group of carbohydrates can react with acids such as phosphoric acid to yield phosphate esters and these are intermediates that are found when the glucose is being metabolized. Glycoside formation, on the other hand, involves the two cyclic if the cyclization of a straight chain can either produce an hemiacetal or hemiketal, as the case may be. If it's an hemiacetal, that means the cyclic form is from an aldehyde sugar, while if it's an hemiketal, the cyclic form is from a ketone sugar. Now, their reaction, so these are hemiacetals and hemiketals reactions with alcohol to form they react with alcohol to form the corresponding acetal or ketal. So if you have two monosaccharides which are hemiacetals or hemiketals, the two hemiacetals will form an acetal, while if they are ketals from keto sugars, the two of them will form a disaccharide that will be a ketal. Now, on the contrary, when a cyclic hemiacetal or hemiketal form of monosaccharide reacts with alcohol, the new linkage is a glycosidic linkage. You call that an O-glycosidic linkage mostly, and the compound form is a glycoside. Compound that will be produced is glycoside. So now these are structures depicting either if the anomeric carbon involved in the glycosidic linkage is in the alpha conformation or the beta conformation. If it is in the alpha conformation, that will be an alpha glycosidic bond. But if the anomeric carbon involved in the glycosidic linkage is in the beta conformation, it will be forming a beta glycosidic linkage. Now, the reducing sugars. I stated earlier that all monosaccharides are reducing sugars because they can be oxidized by weak oxidizing agents such as Benedict reagent. This forms the crux of the test for sugars. Almost all disaccharides, with the exception of sucrose and trehalose, are reducing sugars. So sucrose and trehalose are non-reducing because their reducing end or their anomeric carbon that will be involved in the reduction oxidization reaction is involved already in forming the glycosidic linkage. So it is unavailable to reduce oxidizing agents, weak oxidizing agents such as the Benedict reagent. So essentially, glucose and other reducing sugars react with Benedict's reagent containing copper ions to form containing cupric ions to form brick red precipitate. This is the basis of the sugar test, the blood sugar test, until it was discovered that for glucose there is a need for a more specific test that would identify only glucose, which is which has been severally identified to be related to diabetes mellitus. So essentially, the need for more specific tests as there's a need for more specific tests that would identify only glucose. So enzymatic tests are now being used that would oxidize glucose, and then the reactant, the product formed can now be assessed under spectrophotometric methods or techniques. So some of the important monosaccharides are glucose, fructose, and galactose. Glucose is the primary fuel in living cells. Like I said, it is the form in which carbohydrates are being absorbed. The cells, such as eyeballs, have limited oxygen supply, and then they use large amount of glucose to generate energy directly. Dietary sources include the plant starch, the disaccharides, lactose, maltose, as well as sucrose. Fructose, on the other hand, also referred to as levulose, is often referred to as the fruit sugar. It is found in some vegetables and honey. This molecule is an important member of the ketose member of sugars. It is twice as sweet as sucrose. It is used as a sweetening agent in processed food products. It is present in large amounts in male reproductive tract. Galactose, on the other hand, is necessary to synthesize a variety of molecules, biomolecules such as lactose in mammary glands and certain phospholipids, proteoglycans, and the rest. Galactose and glucose, as I mentioned earlier, are epimers at carbon 4, and their interconversion can be catalyzed by the enzyme epimerase. Monosaccharide derivatives such as uronic acids are formed when terminal hydroxyl group or alcohol group of a monosaccharide sugar is oxidized. So I mentioned some important acids in animals to include glucuronic acid. Its epimer is hydroronic acid. There are also amino sugars and then the deoxy sugars that are found in deoxyribonucleic acid. So an important deoxy sugar is the 2-deoxy D-ribose sugar. Now,